Hello, everyone. I hope all of you had a great holiday. So today we will discuss scientific papers from research journals. Uh, Altogether, we will discuss two papers. I have already uploaded both to the Blackboard on the Journal Club papers folder. And I'll give the lectures first. And during the lectures, I will present the background information, experimental design, and also discuss the major figures in the paper. All right. So on December the 14th, we will have an open book exam on these two papers. It consists of 20 multiple choice questions, not 50 anymore, okay? But then the 20 questions, you need to complete them in 40 minutes. So the exam will be distributed the same as previous three exams. You can complete the exam anytime between 4 to 5:30 in the afternoon. Okay, and please make sure that you read and understand the article before the exam. The purpose of my lectures is to help you understand the articles better. So some exam questions will come from my lectures and some will directly come from the papers, right? Also, if you haven't done so, please remember to complete the course evaluation in Access UH online, all right? So first, I would like to briefly introduce the structure of a research paper. Although each journal may have special requirements on how to arrange a research paper, the main structure of a research article is basically the same or very similar, okay? On the left, these are the main components of a research article, and there are four major sections, including introduction, methodology, results, and the discussion, okay? On the right here, it shows other components that need to be included in the research paper, including title, authors and their affiliations, abstract, and others like reference and appendix, okay? Now I would like to go over each section a little bit to let you have a general idea what information should be included in each categories. Meanwhile, when you read the paper, please try to identify each section in the paper. Okay, so let's start with the right column shown here. So the title and authors are basically self-explained, right? A title should be the fewest possible words that accurately describe the content of the paper. For the authorship, it's actually quite interesting. As a trainee, such as a student or a postdoc, you would like to be included in the front of the authorship as much as possible. So the first author gets the most credits, and then the second author, and so on and so forth. So these are the investigators who did the bench works, okay? But as a faculty member, or sometimes known as a senior author, you would like to be included in the last authors as much as possible. So these are the investigators who obtain grant support and generating ideas for the project. Okay, corresponding authors are usually senior authors. Therefore, the authorship listed in the middle are usually contribute less to the project. Okay, then let's talk about the abstract. Abstract concisely states the goals and the scope of the study, where these are not obvious from the title. So more important, it concisely summarizes the results and the principal conclusions. Okay, and then the keywords and abbreviations are used by the indexing and abstracting services. And then acknowledgements provides the funding information and any other people contribute to this work, but not qualified enough for the authorships, such as like article editing, something like that. Okay, But then references and appendix, you should all know about that. Just, reference you already did in your group project, right? So the first paper we will discuss is from a journal named the New England Journal of Medicine, or NEJM, okay? So NEJM is world's leading medical journal and has been published continuously for over 200 years. As I previously mentioned, any journal with an impact factor of 10 or above is a very high cited journal. But then NEJM has an impact factor of 74.7. Okay, it reports many results from clinical trials such as this one where we are going to talk about. So the title of this paper is Use of CAR Transduced Natural Killer Cells in CD19 Positive Lymphoid Tumors. So the senior author of this paper, that's her name, Katoyo Resvani. So this is a professor from the Department of Stem Cell Transplantation at MD Anderson Cancer Center. So the link showing here, this is linked to their clinical trial, and you actually can find more detailed information of this clinical trial, which we will discuss in a few slides. So this is the abstract of this article. You can tell that it is composed of background, methods, results, and conclusion. So this is very similar to a layout of a complete article, except that each session is very brief. Okay, so by reading through it, you basically can get a big picture of this study. I will not read through it, but you need to, okay? One thing I would like to point it out as the red line labeled here. So this is a report of phase 1 and 2 clinical trial. Now let's see the left column for the main body of research article. An important function of the introduction is to establish the significance of your current work. The introduction can start with background introduction, information, and then finish the statement of goals, or as some other people prefer, with a brief statement of the main findings. Okay, either way, the readers should have an idea of where the paper is heading to, follow the development of the evidence. All right. So then the main purpose of the methodology, or sometimes people call the materials methods, this section is to provide enough details for other researchers to repeat your study and reproduce the results. The scientific method requires that your results to be reproducible, and you must provide a basis for repetition of the study by others. All right. And then for the results session, you present your main discoveries by displaying figures and tables. Here you can present and digest the data and then extract important trends or conclusions. Then in the discussion session, you should discuss what principles have been established, what generalization can be drawn, and how your findings compare to the findings of others, and what are additional implications. Everything you can discuss. In the exam, I may provide a few sentences and ask you to identify which sections it should belong to. It may not only limited to the 2 papers, otherwise you can just search and find. Okay. So then, before we discuss the paper, I would like to show you a short video made by Dr. Resvani, and she summarized her work and what her lab is doing in her video.
Natural killer cells, or NK cells, are cells of the immune system. The job of NK cells is to continuously patrol the body to look for the cancerous cells or virally infected cells and destroy them. Sometimes, however, cancer cells can make themselves invisible to the immune system. They may not be recognized by the natural killer cells. So then what we do is we can genetically modify our NK cells. We get our NK cells from umbilical cord blood, which is left over from a baby's birth. We introduce a molecule, which is called a chimeric antigen receptor, or a CAR, in the NK cell. And through this CAR, the natural killer cells now can recognize the cancer cell. Like other cell therapies, CAR-NK therapy has two parts. We give the chemotherapy over 3 days, and the purpose of this chemotherapy is not to treat the cancer, but to make the body ready for the CAR-NK cells that are given to the patient. 2 days later, we give the CAR-NK cells to the patient through their vein, just like you would a blood transfusion. The side effects related to the cells is minimal, if any. Of course, we do see some side effects relating to the conditioning chemotherapy, but these are very easily manageable. Currently, our CAR-NK cells will only work for patients with chronic lymphocytic leukemia, non-Hodgkin lymphoma, or acute lymphoblastic leukemia. However, in my lab, we are working very hard to develop CAR-NK cells to target breast cancer, glioblastoma, and other types of tumors.
As we just talked about, this article is a report of clinical trials. So let's first see what is a clinical trial. When scientists develop a promising drug, tests and treatments are first accessed in the preclinical trial research. So preclinical research is done in animal models, such as mice and non-human primates. Then after preclinical research, tests and treatments have to go through a series of clinical trials before they are approved to be used in therapy. Clinical trials test if the treatments are safe and work in people. Clinical trials have at least 3 phases. Phase 1 trials, they are aimed to find the best dose of a new drug with the fewest side effects. So the drug will be tested in a small group of only 10 to 20 patients. Doctors start by giving very low doses of the drug to the few patients. Then higher doses are given to other patients until side effects become too severe or the desired effect is seen. The drug may help patients, but phase 1 trials are to test a drug's safety. Okay, that's the main purpose. So if a drug is found to be safe enough, it can be tested in a phase 2 clinical trial. So the phase 2 trials further assess safety as well as if a drug works. So phase 2 trials are done in a larger group of patients compared to phase 1 trials. Often, new combinations of drugs are tested. If a drug is found to work, it can be tested in a phase 3 clinical trial. Okay, then phase 3 trials compare a new drug to the standard of care drug. These trials assess the side effects of each drug and which drug works the better. Okay, phase 3 trials enroll 100 or more patients, and phase 3 clinical trials are often needed before the FDA will approve the use of a new drug for the general public. Oh, too much. So these are the information I copy and paste from their clinical trial website from this story, okay? The CAR-NK therapy is to treat several diseases, including here said B lymphoid malignancies, acute lymphocytic leukemia, chronic lymphocytic leukemia, and non-Hodgkin lymphoma. Okay, please think about why all these cancers can be treated with this therapy. For the drug, also showing here, this fludarabine and the cyclophosphamide, and those are the chemotherapy drugs. And here the Mesna is a drug used to reduce the undesired side effects of certain chemotherapy drugs, like showing here these two drugs. So the Mesna has been referred to as called a chemoprotectant. Okay, protectant. I'm sorry, it's a chemoprotectant. Okay, so biological here showing is the name of the CAR NK cells they are using. Okay, and we will specify it in a few slides later. Okay, so this is a induced caspase-9 CAR with the CD19, also co-expressed with IL-15 and transduced cord blood derived NK cells. Okay, so this also this drug AP1903, also called remidusib. So this is a dimerizer agent that acts by cross-linking the molecule called FKBP domain of this caspase-9 suicide switch, and rapidly induces apoptosis or cell death. Okay, so it has this description in the paper as well. So previously we introduced CAR T cells. And if you don't remember what CAR T cells are, please go back and check this old slide shown here. In this study, the authors use CAR NK cells, that's CAR natural killer cells. So they are essentially the same on the construction of the CAR molecule. Okay, the CAR molecule actually the same. But then the only difference is that natural killer cells or NK cells are genetically modified other than the T cells shown on these slides. Okay, but why NK cells? What are the advantages and disadvantages about using NK cells to kill cancer cells? So there's a good description of this in the introduction session about this in this article. So please make sure that you read and understand it, okay? Why they're using CAR NK, not CAR T, all right? Okay, so this is an interesting slide. So it's a good summary of this article. And I found this picture actually in a commentary article online. So the top picture explains the composition of this CAR NK, which consists of CAR that specifically recognize, first of all, CD19 molecule, because CD19 CAR, and engineered IL-15 molecule. Okay, so IL-15 is an important cytokine that sustain the viability and activity of natural killer cells or NK cells, all right? So in addition, an induced caspase-9 is engineered. So what is the purpose of this design? Okay, well, I would like you to find out in the article, okay? Then in this study, they treated 11 patients with a median age of 60. And out of these 11 patients, they find out 8 patients have responded to this therapy, and 7 of them are complete responders, okay? 2 important discoveries they found in this study. One, the high early expansion of CAR NK cells correlates with response to therapy, and you will see the figures later on, okay? And then 2, CAR NK cells persist for at least 12 months, but do not prevent relapse, meaning that the cancer still comes back, okay? We'll see those results later. So the link showing here is a link to this small article. Okay, now let's go through these data. We only focus on the main figures, but not the supplemental figures. So supplemental figures are not going to be tested, okay? In the article, the supplemental figures are described as like figure S1 or table S1, something like that. They're usually supportive to the main figures. And here is table 1. It listed 11 patients included in this trial, okay? You can see that they all have been heavily treated. This is how many times they treated. And one patient here has been treated 11 times. I'm giving you an explanation to several concepts and drugs showing here. I will not really go through it, but the HSCT is the hematopoietic stem cell transplantation. And this concept is important, autologous and allogenic, okay? Autologous means the cells or tissue obtained from the same individual. So basically it's the same person, so you don't have any immune rejection. But allogenic is different. Allogenic means the cells or tissue obtained from a person other than the patient. So either a matched, related, or unrelated donor. So that means they're coming from another person, all right? And then these are the two other drugs that can use to treat the cancer, okay? And also here, something important is that the HLA allogenic match, allelic match. So we learned this HLA allelic match when we talk about the, remember to talk about the kidney transplantation, the first transplantation cases in the United States, okay? But we never talk about this called the KIR ligand mismatch. So what does this mean? I want to spend a little bit time on introducing this KIR ligand match. Okay, so similar to T cells, the killing activity of NK cells is also controlled by both activating receptors and inhibitory receptors, okay? One major inhibitor receptor is called this KIR receptor, known as killer immunoglobulin-like receptor, okay? So KIR receptor recognize the MHC1 molecule on these target cells. As shown in this figure, the panel A here, this basically MHC1 molecule, it has a match with the inhibitory KIR receptor. And then the interaction will inhibit the activation of NK cells. So as a result, the NK cells will not okay, kill this tumor cell. But in contrast, as shown in this panel B here, the MHC molecule present on these target cells are not recognized by inhibitory KIR receptors. That means it's mismatched. So, and then this will lead to the activation of NK cells. It will have this cytotoxicity and have this killing of this cancer cell, this tumor cell. Okay, so now you can tell, in order to enhance the tumor killing activity of this CAR NK cell shown in the article, it will be the best to choose a CAR NK cells that have a mismatch with the patient, because this will enhance its killing activity. Okay, then here comes the table 2 showing in the article. So this table summarizes the adverse events in the 11 patients who received this CAR NK therapy. It is important to note that no this cytokine release syndrome or graft versus host disease were observed in any of the patient or 000, okay? So these two adverse effects are very popular in CAR T therapy. That's what we learned from Emily's cases, that she has to use this IL-6 inhibitors to control his this cytokine release syndrome, very high cytokine release. So these results indicate that CAR-NK is generally a safer therapy than the CAR-T. Okay, so this is the figure 1 reporting the clinical response to this CAR-NK therapy and the post-remission treatments. This is the most important figure in this paper. Basically, it tells you how effective the therapy is. You can tell that these 11 patients have been arranged in 3 different groups, and each group was administered with 3 doses, different doses of CAR-NK cells. And all the patients started with active disease, shown as a gray here. Partial response is shown as green. This patient shows some partial response, meaning the tumors were partially controlled. And then the complete response is shown as a blue, both this light blue or the deep blue, dark blue. And then by counting, you can see how many patients? 1, 2, 3, 4, 5, 6, 7, 8. Actually, 8 patients have experienced periods of a complete response. But then the deep blue indicates here, although it's a complete response, they have this MRD positive that indicates there's a minimal residual disease detected in bone marrow, meaning that small number of cancer cells remained to be detected in patients' bone marrow, in spite that the patients showed complete response. All right, so that's some anti-cancers have been used to control the minimal residual diseases. I have listed the drugs, what's their mechanistic, how their drug action on the slides, okay? So here, this MRD means the minimal residual disease, what does it mean, all right? And then this slide shows figure 2. And this is the persistence of CAR-NK cells after fusion, okay? After infusion, the colored dots shown here indicating the 3 groups of patients. They were infused with different doses of CAR-NK cells. And then in panel A, the copies of vector transgenes were measured by this assay called qPCR. Remember that? So we should know that this technology, quantitative qPCR, quantitative PCR, it means it can quantify the amount of DNA here. So here, the transgene is the CD19 CAR, the transgene, all right? And then, so basically, this assay measures how many CAR-NK cells exist in patient's body as time went by, because it measures the 360 days. That's almost 1 year. After. So in the article, it says a month, which is not correct. So this is actually days, okay? And then this panel B here, try to compare the transgene copies in a different way. So here, the authors group the patients by their response to the therapy. This P equals to 0.02 indicates that there's a significant difference between these 2 groups. So what do these 2 graphs tell you? What are the conclusions you can draw? You should have some ideas now, and then please read the paper to find out if your thoughts are correct or not, okay? So that's all for today. So this paper is pretty straightforward, very simple. And I think after read through it, you will be able to know what is the clinical trial and how you perform a clinical trial and what kind of conclusion you can draw. And then I'll also help you understand how to write a paper and then what the paper, what kind of message you can learn or pass through publishing a paper. All right. That's all for today. I'll see you later on.